Today we will discuss the final figure that is presented in this paper, where they have identified what are the factors responsible for PDAC, metastasis and progression. So although they are not claiming that these are the only factors, but they claim that these factors plays an important role in the PDAC, metastasis and progression. And during their research they have found out that it's the MLKL gene upregulations that will cause this anti-CD47 expression on the tumor cells, and that gives it do not eat me signals, and thereby prevent tumor cells from the metastasis. At the same time, the macrophages interactions with the tumor cells leads to the high production of the cytokines such as CXCR8, and those CXCR8 helps in the upregulation of ICAM on the tumor cells, and those ICAM can interact with the endothelial cells, and then endothelial cells helps in the metastasis or migrations of the tumor cells across the host. So this is what they have so far identified from their research. So in this video they will use those informations to check whether they can suppress the PDAC, metastasis and progressions or not. So first they took this tissues arrays. They use the tissue samples from the patients who are affected with the PDAC that has different levels of MLKL expressions. So for the patients that have high MLKL expressions, they can see that more CD47 productions, and also these markers, the CIH3 productions are also high. The E-Catherine markers are low. Whereas for the patients that has low MLKL expressions, you can see that low CD47 productions, low CITH3 productions and low E-Catherine productions. E-Catherine is the marker for epithelial cells. So cells are not able to adhere to its neighboring cells when the E-Catherines are low. And this is the quantifications of the tissue microarray, where we can see that CD47 is high, where MLKL is overexpressed in the patients, and the CITH3 is also found to be high, and the E-Catherines are found to be lower. And this is the relations in the panel C. They show the relationship between high MLKLs and the CD47, CITH3 and E-Catherine, and we can see that with increase in MLKL there is increase in CD47 and CITH3, whereas there is decrease in E-Catherine. The cells acquire more mesochemical characteristics with increase in MLKL. At the same time, they will also generate more do not eat me signals to prevent themselves from phagocytosis. So after that, what they did is they used the organoid to check the effect of GW and anti-CD47 on the metastasis and migrations. So what they have find out is that when they use this anti-CD47 and MET inhibitors, they see that decrease in the growth of the organoid after 72 hours. And when they use both when they use these MLKL inhibitors and the anti-CD47, they see the further reductions in the organoid volume, and these things indicates that, and the quantifications are shown on the right hand side, where we can see that the volumes goes down to the maximum where these GW and anti-CD47s are used. And this is the fluorescence intensity quantifications, where we can see that anti-CD47s are high in this case, and also anti-CD47s are high in this fluorescence intensity, indicating that there are more CD47s that are expressed. And after that, what they did is they did the experiments on T1M1 PDO, or the patients tumor cells. And in this case also they can see that after 72 hours the tumor volumes goes down in the case where GW and anti-CD47 used. Just to remind you that GW is the inhibitor for MLKL expressions. So all this, both on the combined effect of GW and anti-CD47, we can see that the maximum reductions in the tumor volume, indicating that these two combinations is more helpful to for the reduction of the tumor volumes. The quantifications also we can see that the anti-CD47s are higher. In the quantifications also we can see that the E-Catherine, yeah, this bottom panel is the markers for the E-Catherine. Here we can see that the E-Catherines are restored where GW and anti-CD47 is used, and also here we can see that E-Catherine is restored where GW and anti-CD47 is used, indicating that both the inhibitors will leads to the reestablished, both the antibodies will ensure reestablishment of the reestablishment of these cell adhesions in the tumor cells, thereby prevent themselves from metastasis. And next, what it is, they co-culture this PDO and macrophages. They use the co-culture condition medium for the PDO and macrophages and check the MET levels, which are indicated by CITH3. So they measure the CITH3s and they can see that the MET levels, this is macrophage extracellular traps, are lower in the case of where they use GW plus anti-CD47, and this is true for this PDO and macrophages, and also this KDO overexpressed MLKL and macrophages. And so the culture medium is obtained from the PDO and macrophages, and in the second case the culture medium is obtained by co-culturing KDO overexpressed MLK and macrophages. In both the cases the results are lower when they use, in the conditions where they use GW and anti-CD47. And the panel G is the representations of the tumor volume, where we can see that tumor volumes are lower for this GW and anti-CD47 cases in all the cases that are shown here, where only GW is used or anti-CD47 is used. So the combined effect will cause the combined synergistic effect will lead to the lower metastasis and progressions of the pancreatic tumor cells. Thereafter they do the in vivo studies in this mice model that has the high immune system, or that has the efficient immune system, and when they use these overexpressed MLKL plus GW and anti-CD47, they see the lowest tumor weight and tumor volume, and simultaneously these tumor volumes are reduced when they use this GW and anti-CD47. And for this anti-CD47 they also see the reduction in the tumor volumes, but the maximum reduction in the tumor volume is when they use this overexpressed MLKL plus GW and anti-CD47, indicating that both effects will cause highest reduction in the metastasis and progressions. And these are the panel I is the fluorescence images for this tumor images in the mouse models, where we can see that when they use GW the tumor's volume is lower, however when they use anti-CD47 tumor volumes are further lower, but when they use both of them they do not see any visible sign, or tumor volume is lowest. In this case the tumor volumes are indicating by the red dot. The quantifications of these fluorescence images are on the right hand side, where they can see that there they are checking the liver metastasis. So what they have find out, the liver metastases are lower in the condition where they use GW and anti-CD47 for this. This is the fluorescence intensity of the primary tumor lesions, that these tumors that are present in the spleen, and also the fluorescence intensity of liver metastasis or the tumors that are present in the liver. There we can see that it's the lowest. In both the cases, the lowest signals are observed where GW and anti-CD47s are used. And when they check the survival capabilities, their survival capabilities are high when the both GW and anti-CD47s used, compared to all the other case where individual reagents are used, such as either GW or anti-CD47s. And in the orthopedic models, and C57, the orthopedic models, what they have found out is that in the orthopedic models they can see that when they use either only GW or anti-CD47, they can see that tumor volumes are reduced, but the maximum reduction in the tumor volumes is observed when they use both GW and anti-CD47. And these things are quantified on the right hand side, where we can see that the total tumor intensity is lowest for the case where GW and anti-CD47s are used. And when they check the liver metastasis, what they have find out is that liver metastasis are higher when the MLKL is overexpressed, but they are lower when these antibodies are used. That is when they use the GW, which prevents the MLKL overexpression. And when they use CD47, only CD47, they see that they see that increase in the metastasis, but it is lower than the control. But when they use both the cases, both the antibodies together, both the reagents together, that is GW and anti-CD47, they see that lowest liver metastasis. And when they check the mice weight, it does not affect the mice weight significantly. In this C57 BL6 orthopedic models, mice weight does not affect significantly, so indicating that this antibodies does not have a drastic effect on the weight of the mouse, but they have an effect on the liver metastasis and the tumor progressions. This is what they have presented in this paper. That is, if this GW is combined with anti-CD47, it can suppress PDAC metastasis and progression. So I hope you understood this basic results, or the main results that are presented from this experiment. If you need to understand more about their results in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.